Yeah, we have no, I think there are two left or something like that. We'll talk, there are 200 in March.
So some of them are not existing at all, and some of them are changing, so they don't.
No, we do not have active vimornasal genes, which are the genes for the herpes. We don't.
Yeah, I understand we don't have any active.
Yeah. There is. Oh, yeah. Historically, yeah, we have in the history of life.
So we totally lost some of them, and some of them we also lost, but they're like in the gene, so you can see that we used to have them.
Yes. You see that we used to have them, but not humans. When we were mice. In evolution, yes. Okay, we'll talk. Hopefully, we'll talk about the vimon. There are other types of. So there are actually 4 families. This is the olfactory receptors we talked about. This discovery of Linda Buck, 1000, and there are 2 families of vimornasal receptors. Hopefully, we'll get to talk about that. There's a rather new one, just 10 years ago discovered. They're called the TARS, the trace amine receptors. Very unique family, very interesting orders that they encode. Okay, the last point I want to say about olfactory receptors is that there is large variation within a species. Okay, so if you look at my receptors in my nose and your receptors in your nose, each and every one of you, it's a little bit different because there are SNPs, right? You know what SNPs are? Single nucleotide polymorphism, which will cause small changes in the protein. And the question whether these polymorphisms have any functional impact. And in fact, they do in many cases. So this is the work of a group, in this case, Hiroaki Matsunaki, the one who discovered these shuttling proteins. And what he's doing, he's taking tissue from human beings, and there is a known phenomena that the odor androstenone, if I will give you to smell the odor, most of you will tell me it smells sickening, it's a disgusting odor. But some of you will tell me it smells like vanilla. Okay, so if you do a perceptual test, you'll see some variation in the human population perceptually. And what they found in this nice Nature paper is that when they sequence the genome of the people who smell the androstenone, they can find a correlation based on the mutations. So each and every one of you, if you think that androstenone smells like vanilla, you most probably have this single nucleotide polymorphism in this protein, which is quite remarkable that you make a single change in an amino acid here, and this changes your complete person. Okay? So a single nucleotide polyform in a protein in the nose will tell you if this is a good or a bad smell. So it's quite dramatic. This was some correlation here, but not completely explaining that. And in fact, if you look at evolution, you can find, let's say, olfactory receptor number 750. Sometimes they have an ortholog in chimpanzee, human and chimpanzee, or mouse and rat. You can find them.
What do you mean by ortholog?
Ortholog is that it's the same gene, but in a different, in the chimpanzee, for example. Okay, now what is it? It's not exactly the same. It's, there's some criteria for the genetic profile. But they are basically it's the same protein with very, very little changes. So if you look at orthologs between human and chimpanzees, in this case it's a study of 17 different orthologs, and in mouse and rat, you can see that many of them are actually very different. So they do these assays in the hex cells, in the tissues, and ask which odors are these receptors activated to. Only 6% are indistinguishable between the mouse and the rat. And it's roughly the same protein, but with some modifications. So and in humans, if you do the same analysis in humans, okay, so you take my olfactory receptor number 820 and your 820, and you ask which odors do they respond to. 70% is indistinguishable. You'll get the same profile. But still, 30%, you get either they're hypoactive, they call it hypofunctional, hyperfunctional, or unclassified, or they really define the difference. But this means that there is a huge variation within the species, between us, between different animals. We're expressing different types of receptors in our noses. Yes.
Are these odors that are common in nature? Like the first odor that you showed us, what that sometimes smell like vanilla? Like,
Yeah. So it's a good question. We call this, so in audition we call it the soundscape, or the statistics of the sounds around us. You would imagine that the receptors in birds will have a different odor scale. Fish will have a very different odor scale. This we know for sure. And their receptors indeed detect amino acids that are in the water and not volatile, for example. And different animals would be tuned to different spaces. But we don't, again, people did not really map the space in a very robust way. Certainly not with different genes and doing the screening right, etc. So, again, it's a young field with all its problems. So it does fit to the niche, the ecological niche that the animal lives in.
because like, I actually thought about more. I don't know how it has to be related, but like if there's large variation, then this probably means that having that specific receptor isn't essential for the animal to survive or something like that. Because otherwise, like the body would have incorporated more ways that it would stay the same.
Yeah. So again, it's a spectrum. Okay. So you have smells, at least for mice, that are very important. That have to do with appetite, with food seeking, and dangers, especially. Okay, so for example, there is a component in urine of predators that activates a very strong and specific pathway in mice. For humans, frankly, my nose is now clogged. I can't really smell very well, and I can pretty. If I'll be blind or deaf, I'm in much big trouble.
So, for example, that a thing in the urine that causes the mice to be activated. Is there a receptor for that also largely varied inside the mice?
I don't know the answer to this question. Again, people who are doing the genetic comparison would know better than I do on this stuff. So I don't know the answer. To profile the predators and what kind of compound, we know very little about it. Really very little. We used to work on, in my postdoc, we used to work on urine. So you would go into the lab, it was like a restroom in the bus station or something. So we spent a lot of money, not me, they spent a lot of money on vacuums in the and we would buy urine from the zoo. Lions, giraffes, etc. We had a huge closet full of urine and supposedly predator odors. Some people even in agriculture they use fox urine to put in the fields to have the rodents escape. So it's potent. One component which is very potent is this TMT. So it's called TMT. It's very strong, very potent. Yeah.
The perceptual thing is not necessarily relevant to the survival, because it's like one sees urine as the vanilla and another one as something else. It's still both, it's urine. So not sure what does it say, these perceptual differences.
Again, I don't think that each one receptor. So first of all, if you knock out the whole olfactory system from a mouse, he's in deep shit. Really, he's in real trouble. He cannot survive. They will not find their mother. They use their sense to find the nipples of the mother to get milk, and they have to have it for survival. They can be blind and deaf. For us, it's a different story because we rely mostly on hearing and vision. But now, if you talk about a single receptor, specific receptors, I would say is not critical. You have so much different aspects of life which are covered by this that. So maybe so we'll. So maybe we'll jump a little bit ahead. Yeah, a few years ago, this was in Ynet. Okay, this was in Yediot Achronot Science about new mice that are no longer fearful from cats. Okay, so a Japanese group knocked out of the 1,000 genes, a subfamily of 300 genes, only these 300 genes, and they claim that this is a pool of the receptors that have to do with fear. And these mice no longer fear from the predators. Okay, so yes and no. I mean, one receptor won't make a change, but we do see that it can have an perceptual impact. Okay, so like we saw in the SNPs story. And the more dramatic one, so the SNPs was I think in 2008.
Like that mouse was, like the genes they knocked out were only factory genes?
Only the 300 or factory genes, yes. Okay, so they knocked the full family. They took up a chunk of DNA, probably from chromosome 11, where there was. I don't know, just kidding. I don't know the exact place of each gene. In humans, the most dramatic, the most extreme example is an olfactory receptor called OR5A1. That's the name of the receptor, and this has been de-orphaned. So this is the odor called beta-iodine. And beta-iodine is used in the food industry a lot, in chocolate and things like that. And what they showed in this paper is that genetic variations in this gene will determine the food preference of the people with the specific gene. So this is the most extreme example that I know of to answer your question. So if I go and I map your olfactory receptor number 5A1, I will tell you probably if you like McDonald's or Burger King or pizza, you prefer pizza or chocolate. Okay? So that's the most dramatic example of the... Okay, few more things about the discovery of Axel and Bach that changed the world. Okay, so now we know that we have 1,000 genes in the genomes that are expressed in the epithelium. Well, Linda Bach went to have a position in Harvard. A new young scientist comes to do a postdoc in Richard Axel's lab, a guy named Peter Mumba. Now he's in Germany, and he makes transgenic mouse. He goes to one receptor out of the 1,000 and expresses GFP in a knock-in manner. So all the receptors will be expressed normally, but will also express GFP. And he slices the brain, and to his amazement, he sees two things. One, so this is the epithelium, okay, with the turbinates on the, and he sees many neurons here expressing GFP. And he counts them as part of the population, and he sees that only 0.1% of the cells are actually expressing GFP. What does that mean?
Receptors only that percent of the issue.
Which means we have 1,000 receptors in this, generally in this tissue, that are expressed. You take the whole nose, you grind it in the machine that tells you how many genes there are, 1,000 is the number. Now you tell, I'm going to mark one gene, and you see this gene only in 0.1% of the population. What does it mean?
Maybe it's uniformly distributed if you have this sample of this. Like,
No, you see that these neurons are uniformly distributed, but if you look here, only there are 1,000 genes which don't express this gene. 999 neurons. And 1 out of 1,000, 0.1%, would express this protein. What does it mean?
it's like you have to co-express another one and see that they don't all both.
Fine, but what does it suggest? It suggests
That maybe
that each neuron will express only a single gene out of the 1,000. So that's one strong evidence for that, which is quite remarkable.
I don't understand the logic of the case that maybe they will only express one.
What do you mean? Now we have cells in the epithelium of the nose.
Yeah.
Okay? They will express maybe 1, maybe 2, maybe 10, maybe all 1,000 genes. Maybe each neuron can be responsible for expressing on its dendrites all the 1,000 genes, maybe 500 genes, we don't know. Based on this data, statistically, it suggests, it doesn't prove because we didn't go on all the neurons and all the receptors, but if you find your receptor only on 1 divided by 1,000, 1/1000th of your neurons express your GFP, it probably means that each neuron expresses only 1. Furthermore, what he did is he picked up the GFP neurons and found that they indeed express this M71 receptor. Well, that's not surprising, but he found that they express only this out of all the genes. And when you pick up randomly neurons from the epithelium, you find that it expresses only single mRNA species from the 1,000 genes. Now, this by itself opened the whole field in genetics about gene regulation. How does a neuron know to express only one gene from the great options that he has? Why not 2? Why not 3? It has implications for function as well. Okay? So this was a great discovery. That's one thing. And the second image was when he looked into the axons of these neurons, which was also quite remarkable, and I just want to show the image. So this is another mouse. These are the neurons. It's a knock-in mouse, expressing not GFP, but some kind of a black marker. You see these cells in the epithelium. And when you look in the central nervous system, all the axons of different cells that express the same receptor, they find each other like friends, and they coalesce in bundles, all the receptors, all the axons. Okay? So he made 2 huge discoveries, Peter Lombard in Axel's lab. One is that each neuron will express a single mRNA from the olfactory receptors, and that all the neurons in the nose eventually converge into a very tiny spot in the central nervous system.
So did he act like that wasn't proof for that? It was only suggesting that each neuron only expresses one receptor.
No, that is very, very strong evidence. That's proof. As close as you can go to get to proof.
At least, I don't know, did he do it for different receptors as well, not only this one?
Yeah, so what you do is he made the M71, he made this other receptor, and each neuron that expresses that will express what he asked him to express, of course, what he's tagging, but also when he looks if there's expression of other receptors, none. Now we know the mechanism of shutting down. Once it's a random, it's like throwing dice. One is expressed. Once it's expressed, it's starting a huge genetic shutdown machinery, specifically in the OR genes. Also a big field in genetics that this started. The other field that this started is axon pathfinding. So again, I don't know how deep you did developmental neuroscience, but one major challenge of the nervous system, to build a nervous system, is axons which travel long ways. They have to find their way where they need to go. And here it's a dramatic example of about, 20,000 neurons or so in the epithelium, which sit very dispersed along the epithelium, but still these axons find one another to go into the central nervous system together as a group. Yeah.
As I understand, in an adult mouse, a basal cell will eventually develop into a neural cell, right?
Yes.
Randomize one of the 1,000 receptors and express it.
Yes.
Then send
the axon to a specific place.
One of the 1,000 right versus.
Wow. Welcome to neuroscience. Absolutely. Not that I'm against any other tissue, okay? But you look at the epithelium of the skin, all the neurons are, they're like soldiers. They stand in a row, epithelial cell. And this is exciting in biology. You know why it's exciting in biology? If you took Introduction to Biology, you would know. But so these cells are bipolar. I mean, one side is, they have the cilia, I don't know, to the gut or whatever, the airways. And the protein synthesis in these very simple epithelial cells has to be, specific proteins have to be on this side of the membrane, specific proteins shuttled here, etc. But these are epithelial cells, little tiny 2 microns. Neurons are amazing, okay? Neurons are, this is just the beginning. They have axons and dendrites, and they travel distances, and they find each other, and synapses on. So it's a really remarkable challenge, the nervous system, in that sense. And the olfactory system has been a model for gene regulation and also axonal pathfinding. Okay, let's go back.
So, just a question.
Yeah.
So there is the convergence to stem cell.
Mm-hmm.
Okay. So there might be thousands of those.
About 20,000.
About 20,000 targets for convergence.
No, 20,000 neurons in the epithelium.
Okay.
They coalesce into a single spot.
So there are about 1,000.
There are 1,000 targets.
So these are the basics of the smell space.
Fantastic. But what I'm saying is that you didn't need electrophysiology. This is what I argue with Haggai Berman. Suddenly, the molecular biology gave us a pathway of organization, of functional organization, into the circuit. Okay, so that was my argument. Of course, if you really want to understand fully, you need to understand neural codes, spiking, if they spike one after the other, temporal code, etc., rate code, space code. I don't say it's not true, but I'm just saying I'm from the UN. We have to. I come in peace. Okay? The molecular part is also powerful and important to the study and understanding of the brain in general. Okay. And I'm not disputing or refuting any other field or method, and that's important to mention, okay? Because you find this very, very often in a rigorous community like ours, okay? That people will tell you, electrophysiology is shit, fMRI is crap, behavioral neuroscience is not. So, we have to be a little bit more humble with how we refute and disregard other fields and people. But you will still find it a lot, especially in LSEC, especially in the retreat. Strong arguments. You weren't here last year, right? So I'll spare you the gossip. Next, when you look at the epithelium and do many of these mice, okay, so you saw one mouse expressing receptor number 71 out of the 1,000. If you do it to other, what they find is that if this is the whole epithelium, a single receptor type will be expressed in a zone in the epithelium. Not in the whole epithelium, but still widespread in large distances. But if you do it for another receptor, also you will see only 0.1% of the cells expressing it, but they will be expressed in another zone, or roughly there is 4 different zones, 250 receptors will be expressed in the different zones, and to this date we don't understand what is the role of the different zones in the epithelium. Okay? So, but nevertheless there is this organization. Probably it's for developmental reasons, but so these receptors develop earlier, and then these, and the substructure of the system. But we'll talk about brain maps later on. This is actually the dorsoventral direction. I don't know if it tells you something. It's important to you? So if you look at the epithelium, it's not very obvious actually. So the epithelium is huge, something like this, and the stripes are like that. Okay. So and then one type of receptor, you do a knock-in mouse. Let's say you have $100,000 to make a knock-in mouse. Although today with the CRISPR you can do it in $2,000. You'll find that your neurons are here, and they will send their axons behind the board is the olfactory bone. So let's do that. Let's go to the olfactory bone. This is the olfactory bulb in mice. This big bulge in the front of the brain. This is above is the cortex, cerebellum, brainstem, etc. And as you can see, it's a big chunk of the mouse brain. In humans, this, of course, as you know, what happened in humans in evolution is that we gain mainly cerebral cortex, so we can do not smell odors, but do mathematics and Neshesot Aleph and Bet. And the olfactory bulbs are these tiny little structures which have been squeezed under the cortex. Okay, this is the first station in the central nervous system of the receptors. Today we know this is again this cut in the face of the rat in the middle. What I told you about today is the olfactory epithelium, this huge structure. Okay, and the neurons here send their axons to the olfactory bulb. But we know there are